Welcome back to Human Anatomy Online. I'm Professor Klein, and today we're talking about the brain and the ear. I'm done playing my piano and ready to help you all learn the brain and the ear. To start, we've got to get to the brain itself, and to do that, we're going back to the skull cap. What's another name for the skull cap? It's the calvera. So that was a question from last PowerPoint. Make sure you're reviewing that. But we take off the calvera and we can see the brain. Now this is only half of the brain here. I've removed the other half, but as you can see, it sits within the skull. We've gone over the skull, so we're focused on just looking at the brain itself. Here we can see the cerebrum, just the outer kind of part of the brain. And really, this is one of the main features that differentiates human brain from an animal brain, is this large, developed cerebrum. So as we go through, what you'll notice is a lot of the words sound and kind of are spelled very similar. So cerebrum, the 4 different lobes that we'll look at, is different from the cerebellum, which would be this kind of posterior structure right here. Also note that oftentimes there's a word to classify multiple structures. For example, the diencephalon is the thalamus, and I'll draw this here, thalamus and the hypothalamus. So number 1, number 2, 1 would be the thalamus, 2 would be the hypothalamus. Together, they equal the diencephalon. Furthermore, something like the brainstem would be similar. It's got the midbrain in orange, the pons in purple, and then the medulla oblongata in green. So be able to list those out as well as each individual structure as well. And looking at the 4 lobes, if we were to look at it like this, most oftenly the frontal lobe will be red or pink or something like that. It's just the color they seem to pick. The parietal lobe back here, temporal lobe in blue, and then occipital lobe in green. If you learned the skull really well, then these should make sense because they're exactly lined up with the skull bones. Now this is a lateral view. Be able to tell me what is posterior and anterior based upon the fact that the spinal cord and the brainstem points posteriorly. Frontal lobe, more anterior. Here's another way to look at the brain, and it's a cross section. So if we were to take the brain like this and cut it straight down like that, you tell me in our small groups what cut is parallel to this plane, or what plane is this cut essentially through in our small groups. But for now, just know that's deep within the brain that we're looking at it. And here we can see the corpus callosum, which connects the 2 hemispheres of the cerebrum. Deep, so it's kind of indented down in here in yellow. But notice there's also something called the longitudinal fissure. That's this line right here. It actually is referring to the indent. So notice that how it goes deep within the brain. That one that runs long ways down the brain is a longitudinal fissure in line with the sagittal plane. So here's a superior view of it. And again, we're looking at an aerial or superior view. The corpus callosum, you can see because the longitudinal fissure has been pulled apart so you can see deep into the brain itself. Back to the cerebrum. We'll focus heavily on the cerebrum as it has lots of functions within these areas. Labeled again, you can see here, but on this slide I want to focus on the gyri and the sulci. So notice how the brain has these folds and then these indents. So a fold would be like this area here, almost like a mountaintop. But in between the mountaintops, you've got a valley. So these indents would be the sulci or grooves. Very important to know those, because those will tell us quite a bit about the functioning aspects of the brain. In addition to knowing the general anatomy of the brain, you'll want to know the general function as well. Here's a summary slide with the general functions. You can see the parietal lobe deals mainly with sensory information. So you touch something, and your parietal lobe tells you what you're touching. Is it light? Is it sharp? Is it painful? That information is being processed in the parietal lobe. Occipital lobe, talking about vision. Temporal lobe, mainly auditory information, language, and a little bit of perceptual judgment. And then lastly, frontal lobe, lots going on in the frontal lobe. It initiates motor activity, speech, conscious thought, personality, logic, decision making, kind of that higher level thinking. So if you want to maybe impress somebody and give them a compliment, you could say, instead of saying they're intelligent, you could say, wow, you have a really developed frontal lobe. So kind of an anatomy compliment there if you choose to use it. Here's another great shot of the brain, or lateral view, where we can see the gyri and the sulci. A couple famous ones that we want to note is the central sulcus. So the central sulcus is this sulcus that essentially runs from the top of the brain almost all the way to this other line here. This other line is called the lateral fissure, dividing temporal from parietal, occipital, and frontal. So start with the brain that's got all the colors on it, but then work your way to the brain where you have to differentiate those areas yourself. Now, I said the central sulcus was famous. It's famous because it runs all the way from the top to the bottom, but it also divides the frontal from the parietal lobes. And two other famous gyruses. So we can see the pre-central gyrus as this section right here, it's in purple, and the post-central gyrus in yellow. The pre-central gyrus, part of the frontal lobe, is going to deal with voluntary motor movement. So when you think about contracting your muscles or moving, it is initiated in that pre-central gyrus. Post-central gyrus, we're talking about somatosensory information. So again, sensory information traveling from your body up to your brain to be processed. Couple other areas to note. Broca's area is one of them. Broca's is in the frontal lobe, and I've kind of put a circle here, but the secret is neuroscientists don't exactly know specifically where it's at, but they know it's in this area. And Broca's area is responsible for speech production. You've got to have speech and the word production or producing speech or something like that in your answer on the exams, you can't just say speech, because there's another one called Wernicke's area back here in the parietal lobe that deals with speech comprehension. So Broca's is speech production, Wernicke's speech comprehension. Something I didn't mention earlier is that the brain also has gray matter and white matter within it. Now, the gray matter and the white matter, gray is superficial, white matter is deep. That's the opposite of the spinal cord. On to our next slide, and I love this slide because this slide does a great job of separating the sections. Now we can say the forebrain is another section, which is a combination of the cortex and the diencephalon. So if I were to draw a line, it would look something like this. This would be the forebrain, with this area here as the diencephalon, thalamus, and hypothalamus. And the thalamus, hopefully you've been reading on this one, it's the relay center of the brain. So if I'm coming in here, thalamus would relay things to the rest of the brain. So it kind of takes things in and then takes them to different areas that they might need to go to. Next up, the cerebellum. So different from cerebrum, the cerebellum is hanging out down here. It's cortex-like, but it's a coordination of the motor system. So your ability to walk or run or do things that are coordinated, and also motor occurs in the cerebellum. And then lastly, the brain stem. We mentioned it was the midbrain, the pons, the medulla. You can see a great picture of it over here. The midbrain's a tricky one because it's kind of just wedged in between the pons, which is much more prominent, and the diencephalon. But do know there is something called the midbrain in there as well. And the further down the brainstem you travel, the more primitive the functions become. So we're talking about autonomic things like respiration and heart rate and blood pressure, and things that just kind of happen automatically in the body occur or are produced from the brain stem. And before I go to the next slide, I do want to note that down here, the hypothalamus is different from what we'll look at later, the pituitary glands. So pituitary glands are different from the hypothalamus. They are connected, but different structures. And here we can zoom in on that. So we've got a zoomed in thalamus, and you can ignore the interthalamic or thalamic adhesion. That's just kind of an area that connects the thalamus on one side and the other hemisphere. But really what we're looking at is this thalamus here with the hypothalamus as, I don't know, what would you call that? A trapezoid, a rhombus, just kind of an odd four-sided shape that traveled down towards the pituitary. Now you'll notice pituitary has a fancy scientific name that you definitely will want to use, and that's hypophysis. Hypophysis means both the anterior and posterior pituitary. There's a front one and a back one. Together they're called the hypophysis. Let's look down below at this picture, and what we can see over here is that the posterior pituitary, this one, is the one that's actually connected to the hypothalamus. So let me put an H up here for hypothalamus. It would come down and attach to the posterior pituitary. Now there's a word I want you all to know, and it's the word infundibulum, super small. You'll see it multiple times, but the infundibulum is the bridge between the hypothalamus and the posterior pituitary. The anterior pituitary is right here, and it's kind of latched on to those other structures. Anterior pituitary is technically not part of the brain. Oftentimes we kind of put it together, but as you can see, it's latched on to the posterior, but the posterior is what comes from the actual brain itself. Quick question before we move on. The question is, the pituitary or hypophysis sits within what bony landmark of what bone in the skull? So take 10 seconds, but you tell me the pituitary gland sits within where within the skull? All right, answer coming at you here. The answer is the sella turcica, which is part of the sphenoid bone. So sella turcica, this area right here, hopefully you've been looking at it, the pituitary would sit right within that. And it's a nice kind of caved in area where you could fit something very safely. All right, I put a little blue pen in there. The blue ball is representative of the pituitary gland. You can see how it sits safely. I'm trying to tap it, and I cannot get to it. Last thing to mention on the pituitary gland is that in human physiology, you'll talk about the different hormones that are produced or released from certain areas, with the pituitary gland being one of them. We'll talk about a few, but you'll need to know those in human physiology, and especially if you want to go into medicine or endocrinology, or hormones affect many, many different things in a diseased population. Moving forward, we've got the diencephalon again, focusing this time on the thalamus. And again, we call it the relay center of the brain, or the coordination center for a majority of the ascending and descending connections. So remember those ascending connection or pathways and the descending connections pathways from the spinal cord. Well, they come up the spinal cord to the thalamus, and then they've got to be sent to wherever they might go. So if it's sensory information, we're coming over to the parietal lobe. Let's say it's something more for the frontal lobe. So again, just sending it out to wherever it needs to go. And as we continue on with parts of the brain, we're going to talk largely about where these cranial nerves are coming off of. For example, in the midbrain, the anterior part here, we've got the ocular motor and the trochlear nerves that branch off of that area before you get to the pons. So now looking at the pons, we can see a distinguishing feature about the pons is this bulge. So this part that kind of sticks out further than the medulla, further than the brainstem, right in here from a lateral view will let you know that this area is the pons. It's also anterior to the cerebellum, and that's important because it's a track to connect back to the cerebellum. You also want to note on the cerebellum itself this intricate meshwork or different divisions called arbor vitae. So these white parts of the cerebellum are called the arbor vitae. Furthermore, haven't mentioned this yet, but there's actually some hollow spaces within the brain itself called ventricles. So we studied sinuses in the skull, and those were hollow spaces in the skull. Hollow, empty spaces in the brain is a ventricle. So right in here, this triangular space, that's the fourth ventricle. There's a few others we'll hit up, but I just want you to start to think, okay, there's some actual structures that you can touch, and then there's also some spaces which are the ventricles. Do note the pons has the trigeminal nerve coming off of it, huge cranial nerve that we've seen before, as well as the abducens, facial, and technically that's all, but again, remember A, F, V, America's Funniest Videos, abducens, facial, vestibulocochlear for these three coming off pretty much the base of the pons itself. Now we're on to the medulla oblongata. It's all right if you just say medulla, that's most often how you'll hear it. But with the medulla being inferior to the pons, but superior to the brainstem, here we can see those divisions. Focusing in on the cranial nerves, vestibulocochlear, glossopharyngeal, vagus, and hypoglossal, but you can see that A, F, V, number 8, is the vestibulocochlear right here, but then it becomes the glossopharyngeal, the vagus, and then hypo, meaning underneath, but it's not up here. This is technically underneath over here, number 12. So notice they've skipped it. They went from 10, 8, 9, 10 to 12. Think for a second. If this is 8, 9, 10, 12, what's cranial nerve number 11? 10 seconds. Think about it. It is shown down here. And 10 seconds is up. It's the accessory or the spinal accessory. So spine, spinal cord. It's very close to the spinal cord itself and actually running onto it. Now, remember, accessory deals with the trapezius and the sternocleidomastoid. So those are both lateral or posterior, which is exactly where that spinal accessory nerve is. All right, we're back up to the cerebrum, and again, we're going to make a cut that comes down and kind of cuts it into a slice, much like if you're cutting a loaf of bread and you pull out one slice of bread, put it onto the screen, and that's what we have down here with this picture up top. Obviously, this would be the whole left hemisphere of the cerebrum. And what this is showing is the pre-central gyrus, the motor, voluntary motor part of the frontal lobe, and it's showing what's called a homunculus. Homunculus is pretty much a body map, and what it says is, depending on the amount or area of the brain that has this area to it, that's how much area of that part of the body it gets. And the more area that a part of the body gets, typically the more function that part of the body has. For example, the hand and the fingers they have a lot of functions, right? They can do a lot of fine movements, and that speaks to the fact that they have more area on the brain itself for motor movement, but as well as the face. But something like the hip or the trunk or the legs even, they're much smaller area because, yeah, you can definitely contract those muscles, but not as fine-tuned as, let's say, the face or the fingers. And we've also got the post-central gyrus dealing with sensory information. These are both on the left and the right, but for sake of the picture, they've just kind of shown one on the left, one on the right, but they're on both sides. And here we can see the areas of the body that are the most sensitive have the largest areas on the homunculus. For example, the face and the fingers and the tongue, they take up almost half of this area of the brain. But things like the leg, the hip, the trunk, the neck, you know, areas that you can still feel, but not as clear or not as sensitive, would get less area. So again, this is a body map, but be able to explain what a homunculus is. All right, looking specifically at the cerebellum, before I just mentioned the arbor vitae, A.K.A. tree branches. That's these white segments in here, very intricate structure of the brain, dealing with coordination and balance and kind of putting a lot of different motor patterns together. So when I was playing the piano while holding up the things in the intro, my cerebellum was hard at work to coordinate all those things together so that they logically make sense. Okay, I've paused here for a little bit because I want to give you some time to review anything from this PowerPoint thus far. So take out a blank piece of paper, give yourself 2 minutes, pause the video, and try to write out everything in this PowerPoint, or the main parts and some details that you can. So hopefully you enjoyed that and you got some time to kind of recap and refresh the brain. I do that not just for my own entertainment and fun, but I do that because that's a really important thing to do when you're studying a large amount of material, is to stop and refresh yourself frequently on previous information. So you might do that 4-5 times each video just for 2 minutes, and you will be amazed and shocked how well you remember the information by stopping and kind of refreshing from there. So give it a try as much as you can with these videos. Next, we're on to the ventricular system. So I kind of alluded to this earlier, but we're talking about the ventricles and what exactly these do and why to know them. So first off, the ventricles again are the hollow spaces within the brain that are filled with CSF. So that cerebral spinal fluid is within the ventricular system, and this is going to essentially fill up different areas and allow the brain to essentially float or be cushioned by the inferior surface. So the brain is decently heavy, not super heavy, but decently heavy to where it will push down on the skull and on other structures and could cause some damage. So this cerebral spinal fluid acts as a cushion from those areas. Now, it does a lot more than that. It regulates brain chemistry and removes metabolic waste and nourishes the brain itself. And it's important that there's this influx of new CSF every single day. So every day you're getting new CSF and you're getting about 400 to 500 milliliters of it. It's not a ton, but definitely enough to fulfill these functions. Now, within the ventricles, and here down at the bottom right is a casting or molding of the ventricle. So this would be like if you go to the doctor and you put your foot in the molding and then it comes out and there's this hollow space of where your foot would be. It's kind of the reverse of what this is, showing those empty spaces. Okay, so down below I've outlined the different ventricles for you. There's 2 lateral ones, one on the left, one on the right, but then just 1 third ventricle and 1 fourth ventricle, as you can see down here. Now, the bridge, or what connects the third and the fourth, is called the cerebral aqueduct. I've put that in this color here. So if you're looking in here, that's the cerebral aqueduct connecting the third and the fourth. I've also highlighted with this purple color the interventricular foramen. Now again, remember, foramen is a hole, so this would be representing a space or a hole. Now if we were to jump up here, I could kind of draw these on where that might be so that you can see these things within the brain. Okay, so I've drawn these in, and again, you can kind of see this bottom picture up here. So you've got the fourth, which is right next to the cerebellum. You've got the third kind of right on top of the thalamus, but it's in there. Then you've got that cerebral aqueduct connecting the third to the fourth. The interventricular foramen, tried to put it right where that arrow is, and then the giant lateral ventricles, which would surround both the sides. All right, I know that's a ton of new information on the ventricular system, and there's actually a process we want you to know with this. So take some time with these, but when you're ready, we can talk about the CSF circulation. On an exam, you're most likely going to be asked to track it through the different parts of the anatomy. Now, this picture is okay. I put this in because it kind of shows just the general flow through the ventricle, starting with lateral to third to fourth, and then going to some things called apertures. So apertures, think of it as a hole. And we've got median and lateral apertures to get from the ventricles to where? Take 5 seconds, 10 seconds to think. If you're in the ventricles, where are you trying to go to? And if you've read the slide, then you might be able to say that we're trying to get to that subarachnoid space, back to the meninges, brain, spinal cord, subarachnoid space. Hopefully it's all starting to connect, but that's where we're trying to get to. Here's my favorite slide of that process. So let me draw it on the page for you as we go. And we had mentioned that you produce CSF in the ventricles, but it's technically in something called the choroid plexus. The squiggly red part in here that I'm putting on black is the choroid plexus. The choroid plexus has some really specific cells that produces CSF. I'll have you guys answer that in the professional small group meetings. But we start in the choroid plexus of the lateral ventricle, number 1. Then we're going through that interventricular foramen. Then the third adds more down in here. We're going with gravity, so we're traveling down the cerebral aqueduct to the 4th. Now we're at the 4th. The 4th, you can't go any more inferior after the 4th, right? So you go out, there's 2 lateral and 1 median apertures right in this area. And what happens is then you enter the subarachnoid space. So imagine this 2D picture and you're going through it or through the picture to get to the subarachnoid space. And now we know that is up in here, kind of travels up and around, surrounding the brain and cushioning it as well. But when we're done with the CSF, it goes out those arachnoid villi or arachnoid granulations into the superior, let me check on this one. My brain wasn't working there for a second. It's right on the screen. Superior sagittal sinus, which is the largest vein all the way around here in the brain. So it would drain into there as the final part back into the bloodstream. Whew, that's a lot of steps just to get CSF to the brain, but know those 8 steps for your exam. Very important cells down here, the ependymal cells. Spoiler alert to that question I just asked about where is CSF specifically produced in the choroid plexus. It's the ependymal cells, and these surround capillaries, as you can see right here. So together, those things make up the choroid plexus, but it's actually the ependymal cells that produce the CSF. One quick clinical issue that can occur with CSF and the accumulation or excessive accumulation in the ventricles is hydrocephalus. And as you can see here, this is a very extreme condition or situation, but it's where the median and lateral apertures are blocked. So as you can see, the brain is swelling with CSF. This is not a good situation to be in, but luckily there's a surgical placement, shunt, that can make another hole so that the CSF can be drained. And again, if you're looking to go into surgery or medicine, knowing the anatomy is super important for not only knowing the surgery, but also understanding how you might discover new ways to fix issues in the future. Transitioning out of the brain and back to the neck, because now we're talking about the vasculature of the brain. And let's talk first about how blood gets up to the brain itself. Earlier in some PowerPoint videos, we talked about the carotid artery. Now we know there's a common carotid artery, and there's an internal and external split. So these split, and when they split, the external goes to like the scalp and the dura mater and kind of the more external structures, but the internal carotid goes deep up into the brain. One thing to note about these words on this slide is the brain, it's needy. It's kind of selfish. I mean, it does a lot for the body. Pretty much everything is controlled in the brain, but it takes up 1/6 of the cardiac output of your blood from your heart, and then it takes up 1/5 of the oxygen consumed at rest. Keeping the brain filled with blood and oxygen is vital for someone to stay alive, as well as have the brain functioning. So you might have experienced when you haven't eaten in a while, or let's say you're not breathing as well, you might get a headache, or you might not be able to study or read. And in that case, it's time to take a break, take a 10-minute break, take an hour break, take a break so that you can replenish your body and fill your brain back with blood and oxygen. Here's another great picture of the arteries, and what I'm going to do now is go through and highlight the ones that you'll need to know. So definitely want to know subclavian, vertebral, external, internal, basilar, and facial, as well as common carotid and brachiocephalic. So all those ones that I've kind of dashed, you'll need to know. The other ones, they're great to know, but you will not be tested on those. So benefit of watching the videos all the way through is finding out what is focused on and what is things that you do not need to know for this course. Okay, and up until this point, I haven't given you any videos from my YouTube channel yet. So here's a couple of them. There's a general one on the brain. It uses the colored version of the brain here. And there's also one on the circle of Willis, and featuring Kyle Corbett and Alec Nader, two anatomy and physiology TAs here in the nursing program at OU. But you're probably wondering, what the heck is the circle of Willis? So let's take a look at what I'm talking about here. So as you travel up to the brain from the neck, eventually you're going to get to the base of the brain, and that's what we can see here. And we're just looking at the vasculature, the arteries that travel up there, one being the internal carotid. I'm going to circle those right here. So this is the internal carotid right next to the optic chiasm, this X. And those would be coming up the neck. But the other one, the vertebral artery, comes up the spinal cord. Small groups, make sure you know what the vertebral artery travels through in the vertebrae to get to the base of the brainstem. But at the base of the brainstem, it turns into the basilar. So this becomes the basilar, and let me use a different color here. This is the basilar artery. So vertebral is up to basilar, and then it really enters an area that makes a circle. And we'll zoom in on this, but this circle right here is called the circle of Willis, or the cerebral arterial circle. Why is this important? Well, it's amazing that we have this, because very few other places in the body do we have a circle of arteries. And what that means is you can go one way around the circle or the other way, either direction. And let's say something blocks part of that circle. Well, we know the brain is so vital that blood just goes the other way. So it's like a roundabout that has no traffic laws, and you can just go either direction to get to where you need to go. That's really beneficial in the brain. Another word that I want to define for you is anastomosis. PowerPoint 1, we said arteries go to arterioles, which go to capillaries, which go to venules, which go to veins. That's the normal pathway. But something called an anastomosis is a direct connection between arteries. So the internal carotid and the vertebral, they've got a lot of different connections between them, and this is prolific throughout the body, but an anastomosis connects two arteries with a linkage so that blood can flow more freely. And here's the circle of Willis without the brain on the right, and as you can see here, we've got the structures that I just mentioned. First off, the internal carotids, they've been cut. So essentially, this is where they've been cut at the base of the brain. The vertebrals, though, we can see are coming up, and they flow together into that basilar. Probably shouldn't use red. The basilar, with a few different things coming off of it. Superior, anterior, posterior, cerebellar. So go into the cerebellum. But then notice how we've got the circle, not a perfect circle right here, but you can see the circle of Willis with the posterior cerebral coming off posteriorly, going to the occipital lobe, the middle cerebral going to parietal, frontal lobes, and temporal, and then the anterior cerebral up top. Those are the three different types of cerebral arteries in the brain, but there's two more that we cannot overlook. These two are the anterior communicating, which connect the anterior cerebral arteries, and the posterior communicating, which connect the posterior and middle cerebral arteries. So jump to the picture on the left, and now we can see where the colors line up. So this middle one would be going over to these areas. Again, it's kind of hitting temporal, it's hitting parietal, it's hitting a little bit of frontal, not so much occipital. Occipital would be your posterior. So posterior is coming over, it's hitting that occipital lobe, and then anterior is coming up and doing the medial. So this is a lateral view up here, but down here, this is a medial view. The medial cerebrum, as well as that frontal lobe. Make sure you know not only the circle of Willis and how to name each artery, but where does it go in the brain? You might be wondering why you need to know this. Professor Klein, why? Tell me why I need to know that part of the brain. Well, if you're in a hospital and somebody comes in, they will never say, I damaged my middle cerebral artery. They will never say that. They just don't know that, right? But what they might say is, let's say they damaged their posterior cerebral artery. They might come in and not be able to pass a vision test. Their vision could be damaged. So that symptom, knowing that vision is occipital lobe, and knowing that the occipital lobe is supplied by the posterior cerebral artery, you track backwards from the symptom to the issue, and now you're ready to treat that patient. In your small groups, I want to have you name these 5 blood vessels. So this is a cadaver picture from a website that's approved, and they've got the 5. So take those pictures, place them onto here, and you guys tell me what each of these are in your small groups. Done with the arteries, onto the veins. On the veins, third time hearing this probably, the superior sagittal sinus, this giant area for blood to drain in the brain. And if we draw on it here, you can see it goes all the way from the anterior to the posterior with tons of branches, right? We're not going to learn all the branches, but here are the veins that you do need to know. So superior and inferior, there's an inferior sagittal sinus as well. So I'll draw an S and an I for those, with a straight sinus connecting them. But then it hooks laterally, and we're talking about the transverse sinus. But then sigmoid sinus would be like this part where it does kind of an S curve on both sides. S curve, and we've hit all those, and now we're at the internal jugular. Down here would be the internal jugular vein. It is a pathway, so make sure you know which pathway goes to which one next. Here's a side view of it, lateral view, where we can see the same exact thing, superior sagittal sinus coming down. We've got the inferior sagittal sinus like a giant drain for the body. And then transverse over to sigmoid, doesn't look as S-curved, but this is still the sigmoid, into the internal jugular vein. Lots of other stuff going on, but again, remember just that list that I put in the PowerPoint is what you're responsible for. Over here, it's telling you the sequence. This is blood mapping. Something you'll have to do in the future on exams is map blood through the body step by step by step. Clinical pause, where we pause and talk about a clinical issue that's very prevalent in the research and society today. Essentially, it's an AVM, arterial venous malformation. Now, I've posted a research article from Dr. Corinne Nielsen, who is a faculty member at Ohio University. Here is Dr. Nielsen. She's a Harvard graduate, but she researches full-time the AVMs in the brain. Back to what an AVM is. Well, essentially, earlier we talked about how an artery goes to an arterial, to a capillary, to a vein, to a venule, and then a vein, normally in this pattern, right? Well, sometimes that can fill up and start to twist and pretzel and kind of smash together, and can swell up and cause some serious issues in the brain. Here is Dr. Nielsen's lab website, where if you're interested in working with Professor Nielsen, you can go to this website on the Ohio Biological Sciences website and learn more about what she actually researches and some current news that she's worked with some students and other individuals. Just got a major research grant. So again, I'm very encouraging of getting involved in research and something that you're interested in. So take this opportunity to check out this website of not only Professor Nielsen, but all the other research that's going on at Ohio University in the Department of Biological Sciences. So in addition to checking out all those websites, you'll want to read at least the abstract, if not more, of that article, and be able to tell me the main findings of her article. So in our small groups, I'll have you all report that to me. And that finishes the brain section of the brain and ear PowerPoint video. So check out part 2 as we get into the ear and learn all about the ear for this. But this is the conclusion of this video. I'm going to go play a little bit more piano.